We're next going to discuss a very interesting chemical puzzle that developed as organic chemists began to study carbohydrates in more detail. This is known as mutarotation, and the solution to the puzzle comes from chemical structures that are called anomers. We saw that most carbohydrates are chiral compounds. As a result, they rotate plane polarized light. Recall from first semester that the direction and the magnitude, which is measured as an angle of rotation, with the caveat that it has to be at the same concentration, the same cell length, and the same wavelength, is a physical constant called the specific rotation and is symbolized as alpha in brackets subscript D. D-glucose is unusual because it was discovered that it had two different values for alpha depending on the conditions under which it was purified by crystallization, similar to what we have done in lab. If we form the crystals at low temperature, the crystals have a melting point of 146 degrees Celsius, and they have a specific rotation of plus or clockwise 112. If, on the other hand, we form the crystals at high temperature crystallization, the crystals have a melting point of 150 degrees Celsius, and they have a specific rotation of plus 19. This in itself was puzzling because the chemical substances from which those crystals were formed were always homogeneous and had the same properties. It was further discovered that if you took a pure sample of either crystal and dissolved it in water, the measured initial rotation would gradually change until it reached a constant, unchanging value of plus 52.7 degrees. This property was called mutarotation, muta being a prefix, which means change. The explanation for this was provided once they discovered the ring forms of carbohydrates and the fact that the ring forms are in fact diastereomers. When we look at the two forms of glucopyranose, which is the cyclic hemiacetal ring form of D-glucose, they have different configurations in the OH that is formed at carbon 1. In other words, carbon 1 has different configurations. There is the alpha configuration, where the OH is trans to the tail, which effectively puts it down in an axial position on the ring. And there is the beta configuration, where the OH is cis to the tail and puts it equatorial on the ring. Both of these configurations can be formed from open chain glucose, just depending on the rotation of the aldehyde group when the carbon 5 OH connects. We also saw that this cyclization forming the ring is a reversible process. When we look at the two different forms of glucopyranose, then, these are a special type of diastereomer. They're a type of diastereomer that, 1, is formed reversibly, 2, can interconvert, and therefore these two different forms of glucopyranose are called anomers, and the carbon that changes configuration is called the anomeric carbon. In the aldose sugars, this will always be carbon 1 because this will always be the carbon that forms the hemiacetal. As shown here, the anomeric pyranose forms are in equilibrium with the open form of glucose. This means that each molecule of glucose is rapidly forming a ring, then being reformed as the ring opens, then forming a ring. And if we look at a specific molecule of glucose, we can see that sometimes it will form an alpha D-glucopyranose, which will open back up and potentially form a beta D-glucopyranose. When the alpha form of glucopyranose opens, the open chain glucose closes again either to the alpha form or to the beta form. Thus, if we had a sample of 100% alpha D-glucopyranose, over time that would change to a mixture of both the alpha anomer and the beta anomer. Thus, the explanation for mutarotation is that the crystal forms that we isolate, either at high or low temperature crystallization, are 100% pure anomers. And when they go into the crystalline solid, they are locked closed in the cyclic form. Because these anomers are diastereomers, that effectively makes them different substances. They will therefore have different melting points and different specific rotations. However, if a sample of the pure crystalline form is dissolved in water, it will begin this process of opening and closing and equilibrating. So that what we can see, if we look at this table, is that over time, either sample would equilibrate to a mixture of 36% alpha and 64% beta. The 100% alpha has a specific rotation of positive 112. The 100% beta has a specific rotation of positive 19. The mixture would have a specific rotation that was in between these, as sometimes the light would hit molecules of alpha and rotate a lot, and sometimes it would hit molecules of beta and only rotate a little. So that as it passed through, the rotation would be in between 112 and 19 according to the percentage of each. So our observed rotation value would be 0.36 times 112 plus 0.64 times 19, which works out mathematically to be plus 52.6. This then is the explanation for the mutarotation, and it also provides powerful support for both the structure of sugars, the possibility that they can open and close to form rings, and the existence of stereoisomers, diastereomers of those rings.